Hi, welcome back. So today we'll be talking about DNA, which stands for DNA-deoxyribonucleic acid. We'll be discussing its history as well as its function and structure and cell activities. So DNA is present on all living things on Earth. Its function is to store genetic information as well as regulate cell activity. DNA is extremely long. In fact, there are about 6 billion base pairs in each human cell. So that's what makes you, you. And even just the smallest difference in structure has incredible differences in function. And this is the concept you're going to be seeing a lot in almost all of your biology classes. Structure equals function. So as a basic overview, if you recall, in prokaryotes, they typically have a single circular chromosome, and they lack a nucleus. Whereas in eukaryotes, there is a nucleus, and it typically holds multiple linear chromosomes. So in prokaryotes, most DNA is contained in the nucleoid. In eukaryotes, most of it is located in the nucleus. However, there are mitochondrial DNA or mtDNA and chloroplastic DNA or cpDNA, which have their own genome, and this can be explained by the theory of endosymbiosis. So as we already touched on, DNA is able to store genetic information. If you think about it, DNA is highly conservative. The DNA that is present in your nuclei in your body, that is actually partially passed down from your ancestors generations ago. DNA actually regulates all cellular activities. It lets a cell know when it should make proteins, what type it should make, how much, and what steps it needs to replicate or make new proteins. So now we're going to be discussing a brief history of DNA. Meister back in 1869 discovered it, however little was known of its significance at the time. Later on, Griffith found that there was something called a transforming principle present by injecting different groups of mice with either the dead or live strains of either virulent or non-virulent bacteria. He found that there was something able to transfer its genetic information from the dead dangerous strain to the live non-dangerous strain, killing the mice. At the time, most people assumed that the transforming principle were proteins, not DNA, so it wasn't until 1944 when Avery, McLeod, and McCarthy showed that substance was in fact DNA, not proteins. Later on, Watson, Crick, Franklin, and Wilkins were working with X-ray crystallography in order to determine the structure of DNA. Long story short, Franklin was very, very good at this type of technique. And Watson and Crick ended up stealing her research notes in order to write their paper and publish it before her, without her knowledge. And then, if you've heard of the Nobel Prize for finding the structure of DNA, Franklin did not get credit while the three men did. This is because of two reasons. One, they did not credit Franklin at all for the amount of work that she did to determine this structure, and two, Franklin had passed away a few years prior from ovarian cancer, which is suspected to be because she was working with X-ray crystallography. So if you recall, DNA and RNA are nucleic acids. These are made up of nucleotide monomers. We'll get to the structure of nucleotides in just a second. DNA is a double helix, and the outside of the helix is made up of the phosphate sugar backbone, held together by phosphodiester bonds, which are covalent bonds. DNA's double helix is made up of the two complementary strands, and these strands are running in opposite directions. That's why they're called complementary, and those strands are connected through hydrogen bonding between the nucleotides. So this is the structure of nucleotides. Just to be super, super simple, nucleotides have a phosphate group, a sugar, and a nitrogenous base. And in DNA, those nitrogenous bases are adenine, cytosine, guanine, and thymine. So I thought this image did a good job depicting the double helix structure of DNA, especially in part A. In part B, how the phosphate sugar backbone is on the outside of the helix, whereas the nitrogenous bases are inside, held together by these dotted lines, which represent hydrogen bonds between the base pairs. You can see that the strands are complementary because one's running in this direction from 5' to 3', and the other one's running in this direction from 5' to 3'. As we briefly touched on in the previous slide, DNA does have directionality. During replication, it's read from the 5' to 3' direction, even though the complementary strands run opposite directions. As you might have heard in other classes, in guanine to cytosine, the difference is that there are 2 hydrogen bonds, again represented by the dotted lines, between adenine and thymine, whereas there's 3 hydrogen bonds between guanine and cytosine. So what's interesting, since DNA is made up of complementary strands, if you're given one strand, you can actually predict the strand of the other complementary. This is because you can always predict that A will match up with T, C will match up with G, and the strands will be running in opposite directions. So the directionality is 5' to 3' in DNA. That means that the other strand will be running from 5' to 3' in the opposite way. So now we're going to just do a quick example to check our understanding if we're able to predict the complementary strand of DNA. If you'd like to pause the video right now to practice, great. If not, we are going to start off now solving it. So if it's starting off with 5', you know the other strand's going to be running the other way. So you can write 3' there. Adenine will always match up with thymine, guanine with cytosine, cytosine with guanine, again adenine to thymine, thymine to adenine, and this carries on, carries on, carries on. And know that the direction for this one's going to be 5' since it's running the opposite way as the first strand. This image does a good job depicting how DNA is highly condensed when it's not being used into the chromosomes. Chromosomes are made up of DNA and protein. They're highly condensed, highly organized, and they become partially untangled during DNA replication when the cell needs to read it, and that's this uncoiling right here. Important definition to know, genes are the part of the DNA sequence that code for a specific product, so it can code for either RNA or proteins. And in a gene, there are multiple codons, and a codon is a sequence of 3 bases. So like this would be a codon right here, ACT, and each codon will code for a specific amino acid. So this is a quick analogy. I did not come up with this myself, but I thought it was an interesting way to think about it. So you can think about DNA in general as a movie production. So the overall organism, this guy, he doesn't have to be a person, it can be an animal, a plant, or whatever. You can think of them as the movie. People are made up of cells, and in the movie production, cells are like the actors. In order for the actors to act, they need to read a script, right? So you can think of the chromosome as the script when it's all packaged up and it's being sent out to make sure that people don't ruin what's going to happen in the next season. And then when the script is actually open, it's out of the envelope, that represents when the DNA is partially becoming untangled so that it's readable. And then if you recall, we went over the definitions of genes and codons. You can think of a gene as a sentence in the script. Just like a sentence, like the dog ran, it gets a point across, just like genes are going to code for an RNA or a protein. Genes are made up of codons. You can think of codons as the words in the sentence. Remember, each codon is made up of 3 nitrogenous bases. So you can think of the nitrogenous bases as letters. So the N in the word ran is a letter, and that represents a nitrogenous base since each codon has 3 nitrogenous bases in it. So I hope this video helped improve your understanding of DNA, and thank you until next time.